Good morning to all. Today we are going to discuss about role of enzymes in digestive system. There are lot of enzymes which is involved in the breakdown and absorption of the macro and micronutrients within the stomach, intestinals and pancreas. Among lot of enzymes, pepsin is the main enzyme which is present in the stomach which absorb the protein. First of all, we want to know what does enzymes role in the GIT. The process of making food absorbable through the action of enzymes which is secreted in the alimentary canal. Alimentary canal in the sense our digestive system, that is a GIT tract. Our daily intake food contains a carbohydrate, lipid, proteins and other micronutrients. Among this, the digestion of carbohydrate, lipids and proteins absorbed through the enzyme process. We already said. In these three columns contains the gastric in the sense it is inside the stomach, and pancreas and intestinal. In carbohydrate, amylase is the major enzyme which absorb the carbohydrate in gastric phases and pancreatic, and disaccharidase is absorbed the carbohydrate within the intestines. The main enzyme in the lipid absorption is lipase. It is the easy name, lipid and lipase. Lipase is the present in all the three areas, that is a gastric phase and pancreatic phase and intestinal phase. Next one is protein. Protein in the sense it is a pepsin, PP. So it is pepsin. Pepsin present in gastric phases and other enzymes which is present in the pancreas and intestines. Among this, in this video, we have to elaborately seen in the protein absorption. For that, we want to know what is protein. Protein molecule consists of many amino acids which is joined together form a long chain. That nearly 20 different amino acids that occurs naturally in the protein. Protein is one of the essential nutrients or the macronutrients in the human diet. But it is not all the protein we eat converted into the protein in our body. In dietary sources of protein, it includes the both animal and plant sources and dairy products, fish and eggs, as well as the grains and nuts. Digestion is the disintegration of complex nutrients into a simple, soluble and assembled form, which is the most of the nitrogen in the diet is consumed in the form of protein, because protein contains amino groups and acid groups. Proteins are too long to absorb. The dietary proteins are hydrolyzed into the amino acid by proteolytic enzymes, which can be easily absorbed. Proteolytic enzymes responsible for degradation of protein and produced by the three different organs in stomach, pancreas and small intestine. Enzymes are not acting as a reactant or the main compound. It is act as the catalyst. Catalyst in the sense it lowers the reaction activation energy. It simplifies the energy and workload. In the presence of enzyme, we will get a product soon. Protein digestion in the stomach. Digestion begins in the stomach, and this is the preparation stage, which is compared to the events that occurs in the duodenum. The presence of food in the stomach, it stimulates the G cells of the mucosa of gastric antrum and proximal duodenum to produce and release the hormones gastrin into the bloodstream. The hormone stimulates the parietal cells of the proper gastric glands, which is located mostly at the bottom of the organ, to produce and secrete the hydrochloric acid into the stomach. Thereby, in the proper gastric glands is also used to found the mucous neck cells, that is producing the mucous chief cells that releases the pepsinogen. Next, all of these substances together with others such as the potassium ions and the gastric lipase are present in the gastric juice, which has a pH that ranges between 1 and 2.0. And some protein rich in disulfide bonds such as the keratinine are resistant to degenerate by the low pH, and hence it is difficult to digest in the stomach. Finally, the low pH of the gastric juices activates the pepsinogen, zymogen to pepsin, and the first enzyme involved in the protein digestion. Pepsin is the first enzyme which is involved in the digestion of the protein. Our chief cells already contains the pepsinogen inside that. While we are eating, the food particles enter into the stomach. Automatically the G cells activated, and activated G cells, it activates the pepsinogen into pepsin form. That pepsin is the enzyme which is involved in the protein digestion and absorption. See here in this picture, our gastric acid in the stomach, and the pepsin is converted into the protein into smaller peptides. The protein is the bulk compound. It is a long chain. It breaks the chain into form a peptide. See here the role of HCl in the protein degradation. It denature of protein, convert protein into a metaprotein, which are easily digested and activated pepsinogen to pepsin, and this makes the pH in the stomach suitable for the action of pepsin. Pepsin is the endopeptidase acting on the central peptide which is bound in the amino acid group, belongs to the aromatic amino acid, example phenylalanine and tyrosine and tryptophan. It is secreted in an inactive form that is called pepsinogen. Its optimum pH is 1.5 to 2.2. It is activated by the HCl. Then this different pepsinogens having a different isoenzymes, such as type 1 which is synthesized by the cells of the fundus of the stomach, and type 2 that is produced in all or the region of the organs. All isoenzymes are converted into the active enzyme. Pepsin at the duodenal level, it stimulated the secretion of cholecystokinin to reduce the pancreatic juice. Next one is renin and gelatinase. Renin is the milk clotting enzyme. It is present in the stomach of the infants and young animals. Its optimum pH is 4. It acts on the casein converting into the soluble paracasein, which in turns to bind with the calcium ion, forming an insoluble calcium paracasein, which is then digested by the pepsin. Next one is gelatinase. It is the enzyme that liquefies the gelatin. The end products of the protein digestion in the stomach are protease, peptones and large polypeptides. Our stomach contains the gastric juice that contains hydrochloric acid, pepsin, renin and gelatinase mixed enzymes. Every enzymes which absorbs the protein in the different way. With this, I complete the enzymes which is involved in the absorption of carbohydrate, lipid, protein and other micronutrients. If you have any doubt in this portion, you can comment me at any time. And today's question was, what are all the different enzymes involved in the breakdown of macromolecules, and mention the process of pepsin formation. Thank you for your patient listening.